Hello, today we will be talking about the Meselson-Stahl Experiment. Here I have a quote regarding the experiment by Judith Campbell, Caltech professor of chemistry and biology, and she described it as, "The Meselson-Stahl Experiment has been called the most beautiful experiment in biology for the elegant logic of its deceptively simple design." A little bit of background regarding this experiment was that it was conducted between October 1957 to January 1958 at California Institute of Technology by researchers Matthew Meselson, who we can see here on the right-hand side, and Frank Stahl. Meselson and Stahl demonstrated the semi-conservative replication of DNA, which was initially proposed by James Watson and Francis Crick, but was verified by the pair in 1958. Prior to figuring out the true DNA replication model, there were already three main theories for replication. First, we have conservative replication, which is when two parts of DNA strands are seen to be together after replication has occurred. With the two daughter molecules, one has both of the DNA strands, while the other has all the newly synthesized material. Next, we have dispersive, which is when DNA is chopped up into segments and are then replicated and reconstructed with the old pieces. Finally, we have semi-conservative, which is when DNA, which we know as double-stranded, separates from one another and goes on to copying itself. Here we can see how each of the theories are different from one another. Specifically paying attention to the first generation after replication, you can see the major differences. Moving on to the Meselson-Stahl experiment, they first cultured E. coli in 15-nitrogen medium, which is known to be a heavy isotope, and the DNA that is being synthesized is of heavier density. Once that was done, the bacteria was then transferred to a 14-nitrogen medium, and the DNA was secluded depending on the cycle. Their findings from the first replication is that DNA was only of intermediate density, ruling out conservative replication. Once the second replication cycle was done, two bands of DNA were then found. One was of intermediate density, while the other was of light density, which we can see here in the F2 generation. The importance of the Meselson-Stahl experiment is that it allowed us to have a better idea of the DNA structure. Knowing that DNA semi-conservative replicated, we have a better idea how DNA is preserved and how it is also transferred from parent to offspring. DNA unzips itself and is then separated, which allows for the two strands to act as templates for the newly replicated strand. To better aid your understanding of this experiment, these are two sources I recommend you check out. One is a simulator for the actual experiment itself so that you can visualize everything that is going on. And to use it, all you need to do is enable your Flash player on your device. The next resource is a YouTube video by Bosman Science, and he further breaks down the experiment itself if that's what you want to understand more. Here are all the sources in which I obtained all the information that I used in this video. Thank you for listening, and I hope you have a better understanding of the Meselson-Stahl experiment.